So for this particular presentation, we're going to be talking about one of the efforts, which is restoring habitat for pollinators. And this project was done by Xerces together with Rutgers University, Michigan State, UC Davis, and there was some support from NRCS. And the Xerces Society has been in existence since 1971, doing a really important job of educating us about the role of invertebrates, and in particular pollinators. And pollinators are essential for the successful reproduction of at least 85% of the flowering species in the world. And there's really tiny print there on the bottom that gives a citation for that statistic. In terms of the food that we eat, 1 out of every 3 bites is a plant that needed a pollinator. 35% of them. Now in terms of the nutrition, most of the vitamins and minerals that we need come from plants that need pollinators. So it seems kind of obvious, but it is really important to spell this out to farmers and the general public, that the well-being of pollinators is directly related to our personal well-being and the well-being of our economy. And then it kind of goes without saying for the well-being of our landscapes and the scenic beauty, and for the food web that so many other wildlife species depend upon. So pollinators can include flies, wasps, butterflies, moths, beetles, birds, bats. But we know that bees are the most effective pollinators. They have most of them tend to have special receptacles that hold pollen. They also tend to have hairs that the pollen can attach to. And they display flower constancy, which means that if there's different species in a patch, they will try to stick to the same species, and that ensures that pollen is spread within the species. In California, I didn't update this slide, but the latest count is 1,600 species, and that's cited in Gordon Frankie's book, Bees in California Bees in Blooms. And native bees are very important for the pollination of our crops. Honeybees are important also, but in terms of serving as indicators for the quality of habitat, they're not really very useful. Honeybees tend to forage within 2 miles of a hive, and so the presence of honeybees really is only telling us if there's a hive within that 2-mile radius. Native bees tend to forage closer to their nests, so the presence of native bees is a better indicator of the quality of the overall habitat. So NRCS and Xerces have been very active in trying to encourage farmers and land managers to use native plants to improve habitat for pollinators and beneficial insects. Bees need more than just flowering resources. They need water, nesting sites, and insecticide-free zones. So the question is, if we provide these flowering resources, is this enough? Are we providing the right kind of flowering resources? And are we planting them in a design that encourages the increase in these native bees and beneficial insects? So as with all monitoring, we wanted to develop something that can help us evaluate the effectiveness of these habitat restoration plantings. It has to be something that's relatively easy, quick, and that doesn't require a lot of training or special skill. And something that over time can show us whether the quality of these plantings is improving. A lot of the species that are used in these plantings tend to be woody, so do they reach a point in their lifespan where they're getting too woody and thatchy and therefore needed to be taken out and replanted? Standard methods for collecting information about invertebrates usually include some sort of specimen collection where the insect that you're trying to attract dies. So one of the things that this protocol wanted to find out is whether visual observation gives the same quality of data as an actual specimen collection. So what did they do? These 3 cooperating universities set up 15 to 18 sites in each of the states, They visited each site for at least 80 minutes, and they compared the number of native bees and the diversity of bees and the pollinator plantings to a location close by that had more typical vegetation. They did this 4 times a year for 3 years. What did they find out? Well, first of all, they found out that the number of native bees and the diversity of bees in the restored pollinator plantings was higher than the unrestored sites, and that was to be expected. The other thing that they found is that the number of native bees, the number of individuals counted, was directly correlated to the abundance of diversity. So it had an R of 0.9, so that the more individual bees that were counted, it directly corresponded to the more species that would be present. The other thing that they wanted to find out is the time. So how much time do you need to spend doing this monitoring in order to have good quality data? And they found that after about 5 minutes, they had an R of 0.9, that the quality of the data was really robust. And this was by folks who had some skill at identifying insects. So by collecting and analyzing this information, they came up with a protocol for the bee monitoring. And here are those conclusions that I just mentioned. So the protocol gives us a way to evaluate the quality of habitat and to look at changes over time. So what is the protocol? 15 minutes, ideally twice a year, between the months of May and July, hopefully 2 to 3 weeks between those sampling dates. And the sampling, that 15 minutes should be along 200 feet of habitat. It can be a long strip or it can be shorter sections. The idea is to start with a timer and go to a 3x3 grid and count the number of native bees. Stop the timer, write it down. Other notes can be taken, like the presence of honeybees or the presence of other beneficial insects. It would also be helpful to include what's blooming. Start the timer, move along the transect, again stop, do another 3x3 grid, count the number of bees, stop the timer, make notes. So 15 minutes. It takes a little bit of practice to get that pace, and it also takes a little bit of practice to tell what is a native bee. Because there's a lot of mimicry. This, on this bottom right side, is a bee, is a fly that wants to look like a bee. And then we have a bee, that's the one with the green shiny, that looks a lot more like a wasp. So it does take some skill to be able to tell these apart, and it's not hugely important to be 100% accurate, but it is really cool to develop that skill. And one of the things that we've noticed with farmers is they really, really like to know what it is that they're looking at. They like to be able to tell them apart and know what these different insects are doing in their field. There's resources available to help us with the identification. So this is the one on California insects. And then this California Bees and Blooms is by Gordon Franke and a couple other authors. Encyclopedia of Life has some tools to do different identification. And then this is another book, The Common Bees of California. I don't know if this coupon code still works, but it's 20% off if you order at UC Press. So there's a lot of questions that this monitoring brings up. I don't necessarily have the answers, but what we are finding with farmers is that they need to create these insecticide-free zones. And so that means that they're going to have to increase their reliance on beneficial insects. And for a lot of people, we need to provide good data that shows that if they create these pollinator plantings, not only will it increase the abundance of native bees, but it will also increase the beneficial insects that will provide pest control. This protocol is available online as a free download from the Xerces website under publications, but there isn't a system in place yet to compile the results so that people can compare what they're learning. So that's something that needs to be developed. And then also needs to be incorporated with other monitoring procedures that exist, such as the California Phenology Project, there's Monarch Watch, there's Bee Hunt, the Great Sunflower Project. And then, if people wanted to have help with training on conducting this monitoring, they can contact Jessa Cruz, and her information is available on the Xerces website. She's located in the Sacramento-Davis area. And the training can be a full day and can go very in-depth into telling apart the bees from the wasps and the flies. She brings actual specimens so that people can practice looking at them. And then there's the field component where they can go out in the field and practice the pacing and the counting and the writing down the notes. So this training is available for free for anyone who wants to start using this for your own habitat restoration projects. And also we encourage folks, if they have a garden in their backyard or if they're working with farmers or ranchers, NRCS has a system for taking volunteers. So if people wanted to volunteer and go to a farm where there are these pollinator plantings, an NRCS office could use these volunteers to go out and collect this monitoring. That's what I got for today. Thank you very much.